Oh, you're online, Chris. Uh, Good morning, everybody. This is Town Peterson, and I am happy to receive you in the 19th Global Online Seminar in Biodiversity Informatics. 1 year and 1 day today ago, we did one of these seminars, and during the seminar, I almost burned down my house. Some of you will remember that. But today we have hopefully a calmer event. Today we have Leonard Krishtalka, who's the director of the Biodiversity Institute where I work. Before I give you an extremely brief introduction to Chris, I will put up this. These are ways that you can send your questions for Chris. Do us all a favor and send them right away when they come to your mind. Don't wait till the end of the seminar. And I say that because that way we will have time to pass the questions on to Chris and get you answers while the seminar is going on. So again, you can send it to my email or either of the BioDiv training or BITC emails and Twitter. There's everything. I'll put those back up at the end of the seminar. But again, do send your questions for Chris during the seminar, if you would. Okay, so Chris has enough achievements that I won't list them, but he's been the director of the Biodiversity Institute for 20-some years. We've had a great time exploring this field of biodiversity informatics together. We've sat through innumerable, unbelievably boring meetings. Some of them have been interesting, but suffice it to say, Chris has shown impressive leadership in both leading one biodiversity informatics institution, but also on national and international fronts. Most recently, in his involvement in GBIF and in the JRS Biodiversity Foundation. I'm not going to do any more introduction than that. Chris is going to be talking about grand challenges. We still haven't figured out exactly how many grand challenges, but grand challenges in biodiversity informatics. So it's all yours, Chris.
Thank you, Town. An introduction of my achievements shouldn't take longer than about, I don't know, 5 seconds, 10 seconds. I was going to tell a joke that it's too bad that Peterson's house didn't burn down, because if you saw his house, it would have been a benefit to real estate had it burned down. But that's just a joke. Actually, Town has and his lovely spouse Rosa have done an incredible job on converting an old house into a gorgeous Victorian house.
The good thing is that I have what you just said on tape, so I'll be playing that for my spouse. Anyhow, Go ahead with your seminar.
Thank you. Well, it's a privilege to be presenting, I guess, globally, here in the United States. By the way, for the record, I'm Canadian, but no matter, I still partook in the practice of celebrating Thanksgiving yesterday. This morning, I think we should give thanks that there's still enough biodiversity left to save, and to save in order to continue having biodiversity support the life support systems of the planet. So I have not done this before, and so let's see if I can share the screen and start PowerPoint slides and get on with the seminar. Bear with me. Oh, that didn't work.
Now you're doing well. Now bring up PowerPoint
All right.
and put it into presentation mode.
Are we on?
You're on. I can see it.
Excellent. So, about a year ago, astronomers announced that they had taken the best pictures ever of the Andromeda Galaxy, which is in front of you. And indeed, it is absolutely awesome and impressive. But for me, and of course, for the facts about the Andromeda Galaxy, it's 2.5 million light years from Earth. There's 100 billion galaxies in the universe. And with Andromeda, it'll be 100 billion plus one. The galaxy, like all other galaxies, has hundreds of millions of stars and hundreds of millions of planets. But for me, the critical message is that only one planet in all of these galaxies and all of these stars, only one is known to have life on Earth. So we can speculate about life on other planets, but right now, all we know is there's one planet in the universe that has life, and that's what we should care about. So how do we tackle this, our knowledge of the life of the planet? What are the biodiversity challenges? I'm going to list 5 or 6. The first one is the diversity of life. What do we know about the diversity of life, and how do we get a credible census of the diversity of life on Earth? We haven't gotten very far since the, say, 250, 300 years of the systematic biological exploration of the planet. We haven't gotten very far in understanding the diversity of life on Earth. So here's a simple graph. We know that there's 1.8 million species on Earth, and that has taken about 350 years. But let's say there's twice as many, 3.6 million species, and so we have 1.8 still to discover and describe. How long would that take at the current rate of species discovery and description? Till about 2150. Well, all projections indicate that by 2150, 50% or more of the life of the planet will be gone, and all of us would be paleontologists instead of neontologists, which is fine with me because I was trained as a paleontologist. That's a joke. But what if the estimates of all the field biologists and naturalists of about 18 million species on Earth, some say as many as 50 million, but let's stick with 10 times what we know now, is still to be described. At the current rate of species discovery and description, how long would that take to describe the rest of life on Earth? Forever. It would be gone before we got very far. So something has to happen right now, very soon, and very dramatically, in order to push that curve of species discovery, description, and knowledge to a steep 90 degrees. How bad is this? Town mentioned that I was associated with GBIF. Indeed, I had the privilege of being the chair of the science committee of GBIF for 4 years, and now I'm on the GBIF budget committee. And GBIF is arguably the most comprehensive global source for biodiversity information on the planet. So here's a map of species geographic gaps in GBIF records. These are all GBIF records. Notice Africa, South America, and Southeast Asia. And of course, northern, the Soviet Union Russia and China. I was raised in the Cold War, so it's still the Soviet Union. And of course, Canada. I'm Canadian. I don't know what the Canadians have been doing, but clearly we're missing 70% of the geographic coverage, at least 70, 80% of the geographic coverage of the floras and faunas of the planet. Let's look at specific taxa. Here's from, again, GBIF records. Our knowledge of the birds, not too bad, but still huge gaps in parts of South America, most of Africa, and most of Eurasia. Mammals, worse. Plants, insects, we're starting to drop off now in biodiversity coverage. Again, most of Eurasia, Africa, and Latin America, very poorly known, very poorly represented. Mosquitos, I don't have to draw red circles. You can see where the lights, if mosquito records were the lights on the globe, you can see them slowly being turned off. Sandflies, viruses. This is a very dim picture of our knowledge of the life of the planet after 350 years of the systematic biological exploration of the seas and land surfaces. Let's get even closer, and this is work done by Town and his students. Let's take a closer look, even when we have dots on a map represented through GBIF, and I'm not blaming GBIF for this. They're just serving the knowledge that is served to them from museums, herbaria, other biodiversity institutions, and so forth. But let's take a close look at how good our records really are when we say we have knowledge of the birds. For example, birds are about 80% of GBIF's records. So let's zero in on Kenya. There's 107,000 GBIF records of birds of Kenya shown here. So what's missing? Most of the records are from the southwest of Kenya. So what's missing is much of northern, eastern, and southern Kenya. So even where we think we have a good record, it's not comprehensive at all geographically, and there are many geographic gaps to be filled. What are the solutions? What should we do to increase our knowledge of the diversity of life? Well, as a first order, we have upwards, estimates say, 3 billion records of animals and plants housed in the world's museums and herbaria. They record in one way or another what species it is, where does it live, and when did it live there when it was collected. We need to digitize our biocollections in all of these museums and herbaria strategically and publish these data to GBIF, which would fill in the legacy knowledge of the gathered during those 350 years of the systematic biological exploration of the planet. That would go a long way to knowing what we know up until today, which would then provide a strategic chart of where the gaps still exist that need to be explored and filled. Now, let's take a closer look at all of these records. A good proportion still have the wrong name. Some of them, although they're listed as birds of Kenya, they're not really from Kenya because the data was entered incorrectly. And many of them are not georeferenced accurately and cannot be mapped, leaving about 27,500 records of the original 107 that is really what we call fit for use, fit for biodiversity science research of one kind or another. That's 26% that's ready for science policy. That's terrible. That's absolutely terrible. What should we be doing? What are the solutions to correct this? Well, for the wrong name, let's fix the name data. For the wrong country, let's fix the occurrence data. And there's nothing like mapping it to indicate which of the specimens, which of the dots connected to those specimens need to be fixed. Let's georeference all of our records accurately. And then for the records we do have, 100% of the data will be fit for research and for application for policy and smart conservation. In this species diversity shortfall that we have, to recapitulate, it's time to turn what we now have, which is basically a cottage industry of biotic surveys and of inventories, and very slow description and publication, often 2-3 years in the making. We need to take this cottage industry and make it industrial strength. We need to map all of our current species knowledge, and based on those maps, identify the strategic gaps, both taxonomic and geographic, and design taxonomic surveys and geographic surveys to fill those gaps. We need to conduct, with the specimens that are collected, massive next generation sequencing for quick identification, followed up by deeper research studies. And we need to automate, very important, we need to automate the publication of the results. Publications should take weeks, not years. We don't have years, we only have weeks. If all of the predictions about the rate of extinction of life on Earth are accurate. And of course, we need to serve the data immediately via GBIF and other kinds of portals. The species occurrence and geography shortfall, again, to recapitulate, what are the solutions? Again, map the known species occurrence data. Digitize the global biological collections in museums and herbaria. And you don't have to digitize all 3 billion specimens, but based on the maps of known specimen data and other data, we should design strategically which taxa from which geographic areas should have priority in digitization. And similarly, the specimen data needs to be georeferenced so that they are fit for use for niche modeling, predictive modeling, forecasting, and many, many other kinds of scientific research. We need, of course, to update the taxonomic data and then serve the data via GBIF. Again, this is turning what is now a cottage industry into an industrial strength enterprise. But all of that just deals with eukaryotes. It's like the low-hanging fruit that we are dealing with. In the future, species discovery will be much harder because, as I will describe later on, species discovery is going to become nanobiology, where most of the discovery is going to have to be made in either in the entire microbial world. But more about that later. The second major challenge is the tree of life. How far have we gotten in understanding the tree of life, the evolutionary relationships, the phylogenetic relationships of all of life on Earth? We haven't gotten very far. Here's a simple demonstration of that. There's a known world, and that little red circle is where you are. You're there. Let's start zooming in. It's the animals. You're there. There's Homo sapiens in between the mouse and that amphibian, and each one of these bars is approximately 10,000 species still to be figured out. So there's all eukaryotes. There's humans and chimps and mice, mosquitoes, rice, amoebas. But then the rest of the tree of life that we're just getting started on is microbial life in the form of archaea, and the rest of microbial life in the form of bacteria. Just to show you the magnitude of the problem, the magnitude of the challenge in deciphering the tree of life, and much more about the microbial challenge I'll be talking about in a few minutes. Of course, everybody would love to have the tree of life to be this simple, a Bart Simpson tree of life. I really like the relationships. I don't know if you can see my pointer of fungi to Brillo pads and Limburger and so forth. Very, very funny. But the deciphering the true tree of life is a lot more difficult. The third challenge is deciphering the language of life, and by that I mean the genomics of animals and plants, and of course microbes, which I'll be talking about in a few minutes. It's interesting to me that Escher's lovely painting was only 2 years after Watson and Crick, and I don't know if Escher read Watson and Crick or not. We haven't gotten very far in understanding the genomics of plants and animals. So what should we be doing? Again, massive, strategic, taxonomic, next generation studies and automated analyses, which would then be followed up by other kinds of research across biodiversity science. Of course, if we did sequence most of life on Earth, the total DNA on Earth is estimated to be staggering, 1.2 times 10 to the 37 bases. I don't know what that is, but I know it's a big number. The storage requirements of this massive amount of data is estimated to be about 3.5 times 10 to the 37 bytes. Again, a massive number. Impossible to do now. Why? Because that storage requirement equals the mass of Mercury, Venus, the Earth, and Mars in 2 terabyte drives. Of course, we could also store it all in a side of beef, because DNA, ironically enough, is the most effective and efficient storage mechanism on the planet. And indeed, folks at MIT have managed to store all of Shakespeare's works in DNA. They had a little problem with the recovery from of the data from the DNA. And there are probably a lot of good jokes there. Instead of saying, say from Hamlet, little typos came out there was about a 10% error rate in retrieval, but they're working on it. In any case, we can't do that yet. The fourth major challenge is, of course, how does the web of life, ecosystems, and ecological research. How does the tree of life, the diversity of life, and the language of life all interact to form the web of life and the life support systems of the planet? Well, we haven't gotten very far in that either in 350 years. And we have to get there sooner, because 25% of species are estimated to become extinct by 2050 at current extinction rates. And of course, as we all know, if we remove species, ecosystems crash. It's the famous analogy, of course, is an airplane. Removing species from ecosystems is like removing rivets from the fuselage of an airplane. You wouldn't get on that airplane if you saw a mechanic sitting on a wing, removing rivets, telling you, well, we don't really need that many rivets. The plane will hang together. We need to understand how ecosystems function so that when we do have a better knowledge of the diversity of life and the tree of life and the language of life, then maybe we can apply it smarter in sustaining the life support systems of the planet. Fifth, it doesn't do us any good just to study nature, just to study natural systems, because humans are a part of natural systems. We need to integrate and understand the relationships between natural systems with human systems. One of the worst properties of all natural history museums I know, including the one here at KU that I direct, is that all of the dioramas show nature pristine, untouched by humans. Well, of course, that is false, because of all of nature is touched by humans, and in some cases a lot more seriously than others. We haven't gotten very far in understanding the integration of human and natural systems. As a matter of fact, one of the problems I think we have in studying it is that we don't have a word for the combined human and natural systems. We're forced to say natural systems and human systems. And I think, of course, this goes back to our, what can I say? It goes back to Genesis and the separation of the creation of, quote, natural systems and humans on a separate day. So right from the get-go, in our mythologies, in our creation stories, humans and natural systems are separated, and they still are today. And we need to integrate these two systems from agroecology to urban ecology to energy ecology. And their impacts on biodiversity and ecosystems, our climate and global change, and of course, the reciprocal impacts on our cultures and communities, because the feedback and the reciprocity is ongoing. All of this is captured in the current notion of the Anthropocene, the newest geologic era in which we live. In the Anthropocene, there is no longer a distinction between natural history and human history. They're one and the same. In the Anthropocene, humans are now the most powerful geologic agents that are transforming the planet. Not hurricanes, not tsunamis, not volcanoes, not any geologic force, not plate tectonics, but we are now the most powerful geologic agent. In the Anthropocene, we have realized the ecological limits of human affairs, changing climates globally, changing wildlands to human-managed lands, and of course, causing the massive extinction of species on Earth. In the Anthropocene, as this lovely pieces of art demonstrate, humans are the new God. We create and alter and destroy life on Earth, so that as Goya and Chagoya, the two artists, illustrate, it is the dream of reason producing monsters. What are the lessons that human cultures, and I hope I'm not going to offend folks with deeply religious feelings here, but Easter Island teaches us a great lesson about human and natural systems interacting. Here are the gods lined up on Easter Island. Humans and nature, is the first lesson, are not breeds apart, unlike what it says in Genesis. When the Easter Islanders extinguished their biodiversity, they extinguished themselves and their culture. The other lesson, of course, is that their gods did not intervene. So when one of the current Republican candidates for the presidency, Marco Rubio, said, yes, climate change might be happening. He doesn't believe it, but even if it is, it's God's will, and therefore we should just let it go, because that's what God wants. Well, the gods will not intervene. Six, I think, grand challenge is to realize the ethics of environmental stewardship. The environment, we need to realize, is not a silent, passive backdrop to the human narrative on Earth, not in the Anthropocene. And that we are a biological entity before we are Catholic, before we're Unitarian, capitalist, Protestant, or communist, or socialist, or anything else. All of this, the ethics, of course, in the larger picture, looking at Earth, is to have sustainable nations and a sustainable planet. In this interaction of natural systems and human systems. If these challenges weren't enough, let's go on to the seventh challenge, and that's understanding the microbial world. And I'm going to give credit here to a colleague of mine, Bob Robbins. He and I have worked on this and have submitted a paper for publication on this whole notion. The microbial world is a world of invisible life that was suddenly made visible by metagenomics. Suddenly, and I'm going to repeat this, we realize that eukaryotes are only a minor part of life on Earth. When we talk about life on Earth, we all talk about plants and animals, and we think that's it. That is life on Earth. And yeah, there are microbes there, and there's microbiomes, but you know, that's kind of minor. Well, can these kinds of trees of life deceive us? A lot of diversity is shown up there, and not so much down here among microbial life or prokaryotes. Let's look more closely at an alternate tree based on RNA short subunit sequences, where the branch lengths reflect the sequence divergence. I put a red circle around all the fungi, all the animals, and all the plants. All of classical biology, all Linnaean biology, all Darwinian biology is based on those three tiny branches in this tree of life. So if our goal is to understand biology and the life of the planet, to exploit it for ecosystem function, for pharmaceuticals, for food, for human health, if our goal is to understand all of biology, all of the diversity of life on Earth, the notion that we can do all that just by looking there at those three tiny branches is wrong. Indeed, I would say the new mission, or at least an additional mission, a huge additional mission of every biodiversity institution on Earth is to start looking at the rest of life on Earth, beyond the eukarya. Why? Well, microbes live on every organism, in every organism, and in every single environment, in the air, in the water, and the soils. It's super abundant. Genomically, microbial life composes at least 50% of the life of the planet. In a milliliter of seawater, here's another big number, there are 4 times 10 to the 12th base DNA bases. In a gram and a pinch of soil, there's an enormous amount of microbial life with 2,000 to 18,000 different genomes. Of course, the frightening part is that if you go to a beach and you take a shovel full of continental sand and you throw it into a DNA sequencer, you're going to get 10,000 genomes. If you do it 5 minutes later, you're going to get another 10,000 genomes. And they'll be different than the first 10,000, and on and on and on you go. So what's going on? Lateral gene transfer. This is the first indication that microbes they do not behave the way the rest of life on Earth behaves. They are not a part of classical biology. (65% variation within what we like to call species, but of course they're not species. And it's given the name of a flex genome. Every microbe has about a 65% flex genome. There's a core genome that doesn't change, 35%, but 65% flexes. And then there's the total genome of whatever it is. It's not a species. It could be a network, a colony, something. Then, of course, we know that there's an Earth microbiome in soils and water that likely controls much of ecosystem function. And we are now learning very quickly that there's similar microbiomes within every single living eukaryote, including humans, that controls virtually all of our metabolic functions. So who controls whom? Are we the marionettes and the microbes are pulling the strings? So here's the thought experiment of a metaphorical universe. In the physical universe versus the biological universe. In the physical universe, 4% of all matter is visible and detectable. In the biological universe, 5% of all living matter is detectable genomically. All the rest is undetectable, and most of it is microbial. The remainder we call cosmological, or the physicists call cosmological dark matter. Akin to that, analogous to that. We are calling the remainder, and other people have as well, biological dark matter, microbial life. Cannot be cultured. It can only be sequenced, and that sequence is only valid for an X moment in time, which changes with lateral gene transfer. In cosmological dark matter, this dominates the mass energy of the universe. And I would say that biological dark matter dominates the mass energy and diversity of the biosphere. In the microbial realm, as I've already hinted, it challenges every single concept that we hold dear in classical biology. Let's start with the tree of life. Do microbes conform to our notion of a tree of life as classical biology does, as eukaryotes do? Well, here's Darwin's trees. Notice a single origin for the tree of life. There's Haeckel's trees. Notice a single origin for the tree of life. However, eukaryotes may conform to a single origin tree of life. But because of lateral gene transfer and microbial endosymbiosis, everything changed. And we don't have a tree of life We might have a thicket of life, a bush of life. So we really can't have a tree of life when we talk about microbes. Is there such a thing as an individual organism, which is a fundamental concept in classical Darwinian biology, Linnaean biology? From Aristotle to Plato to Mendel to Darwin, through all of modern classical biology, the notion of the individual organism, of course, is central. But that's not true among microbes. There is no individual. There's no separate development of a body and a germline. There is no body. There's just DNA. And lateral gene transfer means that every individual is continuously varying. So, are eukaryotes even individuals? Is that whole notion of an individual old-fashioned? Or are we all? Is every single, quote, individual among eukaryotes, each of which is carrying a huge microbiome, are we all just holobionts? And in those holobionts, who's really in charge? There's no tree of life. Microbes do not conform to the notion, the concept of an individual organism. It is impossible to define that individual. Is there such a thing as parental heredity? Yes, but most of the heredity, most of the genomic, quote, heredity, is obtained from lateral gene transfer and pickup from the environment, as opposed to direct heredity. Microbes don't, as eukaryotes do, have a stable, regulated genome protected by a soma, by a body. And of course, there is no such thing as a species among microbes. Impossible to define. To understand the world of microbes, this quantum biology, much like in an analogy to physics of subatomic matter, quantum mechanics, we need new theory, we need new concepts, we need new informatics tools to get at these theories and concepts. And we need new informatics tools to capture this and understand this diversity of genomic flux through time. And what are the effects? What are the evolutionary effects, the ecological effects, the phylogenetic effects between this interchange of genes between microbes and organisms? How do they affect one another in feedback mechanisms that affect what we now think we know of classical biology? And what are the feedback mechanisms then in turn affecting ecosystem function back to microbes and back to ecosystem function? We're only just getting started. Let me finish by saying that at the beginning of the 20th century, we discovered that classical mechanics was only an approximation of physical reality. We now know that classical mechanics is only one subset, outgrowth, evolutionary outgrowth from quantum mechanics. And at the beginning of the 21st century, I think we have discovered that classical biology is only a mere approximation of a much larger biological reality. To understand that biological reality, I think we need to recognize that we are fundamentally living in a microbial biosphere. half of Earth's biomass, and most of its genomic diversity. So a top goal for the 21st century, on top of all the others that I've talked about that cover eukaryotic biology, an enormous challenge is to understand biological dark matter and the theory and concepts of quantum biology. Thank you very much. I'll be happy to try and answer any questions that I may have generated. And again, it's been a pleasure and a privilege to address all of you. And I hope that these ideas will generate ideas in all of your classrooms, laboratories, fieldwork, and so forth.
Thanks, Chris.
Thank you.
I'll say a special thanks because I've kind of come to appreciate more and more in recent weeks and months that these seminars are hard to give because it's a bit of a challenge to speak kind of blindly and not know what your audience is.
Yes.
Chris, why don't you finish unsharing your screen? Right now, everybody's just seeing me, which is a little scary. There you go. There we go. There's Chris, everybody. I'm going to share my screen just long enough to give people contact information. Again, email or tweet your questions. For Chris, you can either use my email address or my Twitter address or the BioDiv training email. So Chris, just to give you a question myself, and then I've already got one question from a colleague of yours, but I thought of my question first, so. Okay, I assume everybody has means of contacting. Question for you, Chris. Go back to those more, shall we say, pedestrian challenges of doing business as usual, fitness for use, and you know, even as far out as the tree of life, while you still accepted the idea of a tree of life. How do we pay for it?
I think the answer to that is actually pretty easy. We pay for it the same way we paid for major national and global emergencies and imperatives as they were defined. So when the United States defined an imperative of needing a Manhattan Project, they funded a Manhattan Project. When the United States and the Soviet Union at the time defined an imperative to put humans in space on the moon and then beyond the moon if possible, they started huge space programs. So all it is a matter of defining the biodiversity of the planet as an imperative. And that is slowly starting to gather steam. It hasn't, there's not enough steam, and it hasn't rolled down the tracks fast enough. But in this country, in the United States, more and more money is being devoted to biodiversity research. A lot of it, say the ADBC program from NSF to digitize the most important parts of herbaria and Natural History Museum collections, and surveys and inventories, but it's still a cottage industry. So my prediction is that, you know, we define these imperatives usually when there is a sense of threat, an urgent threat. For the Manhattan Project, it was World War II, and the perceived need to develop a very powerful bomb. With the race into space, again, it wasn't science that was driving that. It was conflict and competition, Cold War competition, and who would own space and who could put weapons into space first in this race in the Cold War race between the US and what was then the Soviet Union. A similar urgency will arise and a similar threat will be seen when there is a major ecological disaster or major human disaster fed by a failure in a biodiversity function, whether it's ecosystem, whether it's a wholesale spread of emergent diseases, where you are one of the leading researchers. An emergent disease that is mediated through zoonotics. Whether there's a massive failure of agriculture because of a massive failure in the survival of pollinators, or the movement of pollinators away from what are current agricultural landscapes because of climate change. It will take, I think, one of those urgent disasters that will make it an imperative for nations to start investing money in understanding the basic life support, what goes into the ingredients of the life support systems of the planet, which starts with the species of animals, plants, and microbes.
Not to bring in a political dimension, but doesn't that depend, at least here in the US, on which way the elections go next year? Think about the example of climate change and climate change denial.
There is climate change denial, just as there was denial about tobacco and denial about DDT, and denial about the ozone hole, until there is overwhelming evidence, and overwhelming evidence that verges on an impending catastrophe. Then whether they are conservatives or liberals, extremists or moderates, the nations will demand it and they will act. I mean, if the crisis is bad enough, it doesn't matter who is president or which party is in charge. They will have to react. It's unfortunate that we humans are, in my opinion, a short-term species where we make decisions based on the quarterly records on Wall Street, the quarterly performances of corporations, 2-year cycles in the election of members of the House of Representatives, 6-year cycles for the Senate, and so on. We're still a short-term species, except for a few things like Social Security. Disasters will, it's for that reason that disasters and crises push nations to the kinds of actions that they have hitherto resisted.
Okay, so here's a question for you from a close colleague, Jorge Soberon, and he's focusing on the latter part of your seminar. He says, this dark matter is very subjective, and no doubt microbes are hugely important and underappreciated. But dark matter in physics is a consequence of a very formal and reliable theory disagreeing with observation. In biology, this is not the case. Can you reflect on that a bit for Jorge?
I will. Thank you, Jorge, for that question. I'll disagree with you. I think it's very much akin to the analogy with quantum mechanics, to understand the microbial world, to understand what is an individual, what is an OTU among microbes. We don't know that. What is a taxonomic unit? Is there such a thing as a taxonomic unit among microbes? They are small, like subatomic particles. They cannot be cultured. You cannot see them. All you do is, for subatomic particles, you get an energy record from an experiment that is momentary. For microbes, you get a metagenomic record that is momentary and is fleeting. In both cases, we are dealing with a time signal problem that is constantly changing and cannot be defined. It is completely impermanent. The difference between the microbial world and the non-microbial world, the eukaryotic world, is a notion of permanence, the permanence of species, the permanence of the individual, and the essential permanence through time and through generations of the genomic record. All of this is not true either in the subatomic world, which is completely impermanent, and the microbial world, which is completely impermanent. And as with quantum mechanics, biologists will need a brand new set of theory and concepts, perhaps likely mathematically based, to understand the entities that we are dealing with, how they behave. Is there such a thing as a predictable behavior? We don't know. And how they, in the quantum world, end up affecting all of the units, the plants and animals, the eukaryotes that live in the classical world of biology. I think that we don't know what we don't know, both about classical biology as affected, and especially of microbial biology, the quantum world.
Okay, well, Chris, thank you for your morning after Thanksgiving.
You're very welcome. No more questions?
No more questions. It's, again, we've always got this little delay where questions will come in the next few minutes, but it's hard to keep things going until then.
So I will just end by saying that my rule of thumb is that nature is not as simple as its observers. All of us are observers of nature. And let's remember that nature is a lot more complex and not as simple as us, the observers, which is great because this shows up how much work there remains to be done.
Okay. Well, again, thank you, Chris, and everybody in the broader community. We usually take the last Thursday in December off from this series, so we'll see you end of January, and I'll be announcing that seminar fairly soon. Chris, thanks a lot, and everybody, thank you for tuning in.
Bye.